being declared, his team pivoted quickly and got a testing lab set up, initially CLIA certified, the clinical lab certification, but we're very proud that yesterday we received CAP certification, the gold standard. That's the College of American Pathologists certification. So we're now a CAP certified lab as well. And I think very quickly we will actually hit 1 million tests. So the saliva test is certainly a much more pleasant option versus the nasopharyngeal swab. Next slide, please. So the evening tonight, A Sip of Science, is really a very pleasant way to learn about science in a social setting. This is our 4th year of Sip of Science. It's actually an award-winning program. We've been named the best way to drink and learn, Best of Phoenix 2020 by Phoenix New Times. Sadly, we're still in a virtual mode this evening to ensure safety, but we'd like to thank the Akatia Restaurant for planning to host the event. And we plan to be there for an event in spring of 2022 and look forward to seeing everybody there. As this is a Sip of Science, we could not have a sommelier here in person, so I have the pleasure of being your sommelier tonight. You have a longstanding interest in wine, Burgundy in particular. Why Burgundy, you might ask? Well, when I was in 8th grade, my father, who was a professor, took a year-long sabbatical and moved our family to Paris. Since French kids drink wine, my father thought I should drink wine too. So I could really have a full French experience. In those days, Burgundy really hadn't been discovered and was quite inexpensive. So that was really the house wine for us. I developed real passion for Burgundy, which persists. For me, the most important aspect of Burgundy is its ability to pair well with food. It's a very food-friendly beverage, and dinner is when I tend to serve and consume wine. So Burgundy is really what I look for. And I always look for a good value, typically wines under $20. For white, I'd like to share one of my favorites with you. The Joseph Drouhin here, it's a nice white without oak, nice hint of lemon. I like things like this Macon Village. It's 100% Chardonnay, as are most white Burgundies. The only other grape found in Burgundy for white wine is the Aligoté. This wine pairs very nicely with a mild white fish like cod or halibut, with a sauce consisting of tomatoes, chopped up with basil, olive oil, and lemon juice. For the red, the Justin Jourdain Bourgogne is really pretty good value. It has very nice earthy flavors. It's 100% Pinot Noir, as are all red Burgundies. Beaujolais, just to the south, uses Gamay grapes. Bourgogne is really a generic wine that can be from any part of Burgundy. As you move up in price and quality, you proceed to specific villages in Burgundy, for example, Mory-Saint-Denis, and then on to specific vineyards, the Premier Crus. And when you get to the very specific subregions, the Grand Crus, you have the most expensive and presumably the finest wines. The earthy things like Jourdain Bourgogne pairs very nicely with chicken with mushroom sauce, as an example. So now let's move on to the main event, the science. We're very fortunate to have with us today Professor Jeff Kordower. Jeff is an international authority in the area of neurodegenerative diseases, especially movement disorders, with a special expertise in experimental therapeutics and pathogenesis. Jeff received his BA and PhD from the City University of New York, as well as an honorary Doctor of Sciences. He's been a past president of the American Society for Neural Therapy and Repair and is dedicated to that disease. He is a pioneer in biological and reparative therapies to treat neurological disorders, and is a founding member of the Scientific Advisory Board and 2-time executive committee member for the Michael J. Fox Foundation. He's performed numerous gene therapy and cell therapy preclinical studies that have been translated directly into clinical trials. So with that, I'd like to turn it over to Jeff.
Thank you, Steve. Very much appreciated. Let me just share my screen here. Can everyone see that? So, first of all, thank you for the opportunity. Many of you don't know that I've only been at ASU since March 1st. And I can tell you there are many people at ASU smarter than I am. There are many people at ASU who are more successful than I am, but I'd be hard to find anyone at ASU who loves their job more than I do, even though it's been a short period of time. It's been terrific. And to build a neurodegenerative disease center from the ground up with the amazing resources that I've been provided and the amazing colleagues that I have surrounding me has just been an absolute thrill of a professional lifetime. So what I want to do is just take a few minutes, just let you know a little bit about me and how I got into neurodegenerative diseases. Because I tell you, if there was one turn left versus one turn right, I would never, ever be here right at this moment. I was at a conference in the Society for Neuroscience where 33,000 people show up every year. And I was in my last year as a graduate student, and I'm walking down the hallways, and I see a sign that says brain cell transplants in a room. I go, brain cell transplants? That's just freaking amazing. How? So we went in the room and we saw these talks about people putting brain cells into to replace damaged brain cells, and they were working. And I turned to my buddy and I said, I'm going to do that. That is like exactly what I would love to do going down the road. So I called up one of the professors who gave one of those lectures, and he wasn't doing anything in neurodegenerative diseases at the time. And I applied for a postdoc at the University of Rochester. A man named was Don Gash. And he said, sure, why don't you come and interview? And I ended up getting the postdoc. And I remember December 23rd, 1979. I went up to Rochester to start my postdoc. And I go, Don, I'm here. Let's go and do these vasopressin transplants. He goes, no, we're not doing that anymore. We're doing Parkinson's disease, and you're the guy. And it turned out that if that never happened, I guarantee you somebody else would be sitting in this chair giving today's sip of science. So with that is just a brief introduction about me. Sometimes you go left and right and it just works out. Now I am the founding director of the Neurodegenerative Disease Center, and I couldn't be more thrilled about it. Before I go into talking,
could you put yours on the full screen mode? We want to see this large in Technicolor.
Sorry about that.
Beautiful. Thank you. Apologies for the interruption.
No, I'm glad you did, because it actually gives me my timing here. So we have four real major missions of the center. One is the obvious one: to prevent, cure, and minimize the disability of individuals with neurodegenerative diseases. But we can't just stop there, because that's not the totality of the process. We have to minimize the burden of the families and caretakers of individuals with neurodegenerative diseases. We have to train future scientists, postdoctoral fellows, graduate students, and undergraduate students, who are, by the way, phenomenal here at Arizona State University, so they can carry on the mission when we've retired, which I'm never going to do, or we move on. And also we'll do it in a way that's highly entrepreneurial, because if you cannot accomplish these goals without binding with industry, because they have the resources to bring you over the finish line. So that's really the mission of the ASU-Banner Neurodegenerative Disease Center. I'm sure in the chat box you can get the links to the websites and learn more. And let me just tell you that I personally will always be available to anyone that wants to ask questions about this science in this space. So that's my introduction. So there it is. Oops. This is what makes human beings human. And it's unbelievable when you hold a human brain in your hand. It's actually an incredible experience. And it's the most complicated brain structure in I don't know why that's happening, in the animal kingdom. You can see we have these folds and these grooves. That's because there are so many nerve cells in this brain that you would have to have a skull this big if you didn't have the folds and grooves. And just to show you, so if you see this is what the brain looks like, is organized. But if it wasn't organized, you have to deal with this. And so that's why you have the folds and the grooves in the brain, to accommodate the skull for all of these brain regions. So this up front is where we think, and here is where we have memories. This strip here is where we have movement, and this strip here is where we have sensation, so you can sense things. And back here is where you can see. If you don't have this area working properly, you can't see. This is where the vision center is, and this is a region called the cerebellum, which is involved in balance. So different regions of the brain do different things through highly interconnected connections. So let me just go briefly over what is inside that brain and how it works. And then we'll get on to talking about Parkinson's disease, and we'll get on to talking about Alzheimer's disease, and also novel experimental therapies that we are extremely excited about. And we're going to go in some of them into the clinic as with ASU leadership, biodesign leadership. So this structure in red is a nerve cell. This is how cells communicate between one another. And you can see these processes that come out, and these processes attach onto other cells, and that's how nerve cells communicate. They are helped out by a couple of different types of cells. This is called an astrocyte or a glial cell, and this helps support this cell. And then you see these white little pieces. That's another type of glial cells, because the neuron elicits electrical properties, and it goes, and these white cells help it go fast down the nerve sending cell, allowing one to communicate to another. So that's how the brain is constructed. And you hope that it's going to work well all the time, but it doesn't work well all the time. And that's why we need to study neurodegenerative diseases. But how important is this really? So I'm going to talk a lot about Parkinson's disease. And Parkinson's disease we're getting to learn more and more complicated, but it was coined by James Parkinson in 1917, actually by watching people walk in the street. They weren't patients of his that he was examining, but there were just people walking in the street, and he noticed these symptomatologies. And then many years down the road, we found out that the difference between a Parkinson's brain and a healthy brain is this region right here. This is what we call a PET scan, but all you got to know is that orange is a chemical called dopamine. And that's the important part for today. So you can see in a healthy brain, you can see all this dopamine, and you can see in a Parkinson's brain, the dopamine goes away. And that's why you have slowness of movement. That's why you have tremors. And that's why you have rigidity, the major motor features of Parkinson's disease. So why is this really important to study? Because by 2040, we are going to have 17.5 million people around the world with Parkinson's disease. And it's growing exponentially. Why? Because first of all, we're living longer. And this is an age-related neurodegenerative disease. And so as the population ages more, the chances of getting Parkinson's disease increases greatly. Now, here's one that's going to probably surprise most of you. People aren't smoking cigarettes very much anymore. And for some reason we don't understand, Parkinson's disease is protective when people smoke cigarettes. I'm not advocating anyone smoke cigarettes, but it just happens to be a biological fact that that's true. And then there are some industrial byproducts. Now, I'm sure everyone has seen the paraquat ads that are on the internet and on everywhere. I can tell you, I'm not going to comment on it because I'm a consultant for the paraquat lawsuit, but that is another particular potential for causing Parkinson's disease. The cost in taking care of these people is going to be economic Armageddon, and the cost in human suffering is going to be incalculable. So we really got to work hard and try and cure these diseases. So you're going to say to yourself, it's going to take a miracle. It's going to take a miracle to cure Parkinson's disease. And could be, if you think about it, maybe it's a more daunting task than what Moses had to do to cross the Red Sea. But I will tell you, in Parkinson's disease and movement disorders, we've already had 2 miracles. I'm going to show them to you now. The 2 miracles are levodopa and deep brain stimulation. Here's a woman with Parkinson's disease. She cannot get out of the chair. Okay, that's not part of the video I'm going to show you. But she's going to be asked to do this finger pointing. And you can see how slow she's going to be. And then the other hand. Now she gets her levodopa, she gets up, she can walk, she looks almost normal. Her hands are a little twisted, but I can tell you that I make all my students go see patients like this, because when you watch a person frozen in a wheelchair and then give them a pill and they can walk like this, it is just one of the most amazing things you could possibly see. But but eventually they get side effects, and that's why we need to do other things. Then with deep brain stimulation, there's a person off his medications, and this is just going to blow your mind. He would fall on the floor if the nurse isn't holding him, and the videographer is going to come in a second and save him. So now we're going to put electrodes into the brain in a region that is overactive, and we're going to stimulate them and shut down that region and watch what we can do. It's absolutely stunning. And it happens immediately. You can even get the benefit to be shown in the operating room. So this is just, we've already had 2 miracles. Let's go after some more. And so one of the ways that we go after it is with transplanting dopamine cells. If dopamine cells are lost in the disease, and then you transplant them and put them back, just like that first day that I saw when I was a graduate student, then maybe that would be good. And so I showed you these images before, a person with Parkinson's disease having very little of this red stuff called dopamine. But then after a transplant in a patient with Parkinson's disease, 1 and 2 years later, we've replaced the dopamine. Now it's been inconsistent, but, and I'll show you where we're going with clinical trials in the not too distant future. So here's another one of the most exciting moments of my career. So I was watching people come into this session, and we're all in the same general age. So you'll understand this reference. I come home one day and I hear the neurosurgeon screaming on an answering machine. Now, when I talk about answering machines to students, they don't know what I'm talking about, but screaming to answering machines that one of the patients in our trial passed away from events unrelated to the transplant. And what you can see here, I want you to know that brown under these circumstances is dopamine. And these are the cells that we put into the brain of this patient and survived for 18 months. And not only did they survive, but they caused more dopamine all throughout the brain of this patient. It demonstrated that decades of work preclinically could be demonstrated in a patient with Parkinson's disease. We published this in New England Journal of Medicine. This is the first demonstration in the history of the world that these cells could survive, spread out their axons, liberate dopamine, and enhance patients' function, patients with Parkinson's disease. But those cells were fetal cells, and there was never going to be a long-term solution. So now, all the groups that are interested in transplants are using stem cells. Now, I'm sure many of you have heard about stem cells. Stem cells can be played around in a dish to make any cell in the body, any cell, liver cell, heart cell, neurons. And there are 2 ways to go about it. One way is to take an embryo, pull out what we call the inner cell mass, put them in a dish, treat them with factors, and make them make dopamine. That is not how we're doing it. In fact, very few people are doing it this way anymore. Now we do it much easier, and also without the ethical discussion about using embryos. My group exclusively uses blood. You can just take a person's blood or skin cells, and then you put these factors in, and Dr. Yamanaka, who discovered this, won the Nobel Prize. And we make stem cells, and then we make these dopamine cells, make them into dopamine cells, and then we put them in the brain with a neurosurgical procedure. That's going to be important just a little bit. So we first had to find out whether these cells would work preclinically. And I'm just showing one slide here. And so these are animals that received a lesion, and look down in the lower left, it's all white. That's because we made a lesion, they have no dopamine, and then we put in these cells, but these cells are too old, so they don't innervate very well. And then as we get younger and younger and younger, you can see the graphs get bigger. The brown dopamine gets more expansive. And I'm not going to show you for brevity, but these animals are functionally normal in these studies. And we have a very logical and orderly survival of these cells based upon their age, and also this innervation part, which is really the critical part, is even better in the younger animals. So these D17 cells are going to be the cells in which we are transplanting in patients with Parkinson's disease. Now, here's one of the very exciting things we have ongoing right now. I was asked by the Michael J. Fox Foundation to write a grant to transplant patients with Parkinson's disease, a special subset of patients. These patients have a specific mutation for which only dopamine is lost. They're really the perfect population to test this. Now, ASU doesn't have a hospital or a medical center, but I was still brought here tasked to have a neurodegenerative disease center that is translational and will help people with Parkinson's disease. And I can't tell you how I've been welcomed by the Arizona community. So this is how the grant has been structured. I'm the PI, I'm the principal investigator, and I'm proudly from biodesign at Arizona State University. Paul Larson, a good friend of mine, has just moved to the University of Arizona Medical Center. Now, the way we want to do this trial is to have people come from their home cities, fly them into Arizona for the surgery and their PET scans. And I got Barrow to allow a University of Arizona medical surgeon to perform the surgeries at Barrow. And also they'll get their PET scans at Barrow, those images that I showed you. And the chemical that makes that brain light up is done at Banner. So we've able very quickly for the center to embrace the neurodegenerative disease space in Arizona. And we're going to let Arizona lead the way in this trial. And then I can tell you once, and if we're successful, we will absolutely do a bigger trial with patients that don't have this mutation, that are just what we call idiopathic normal patients with Parkinson's disease. Now, I wanted to show you one other thing. So I showed you that this is brain surgery. And I once said to a neurosurgery colleague, oh, but it's really minor brain surgery. You really don't have very many side effects from this. And he goes to me, don't you ever tell me that. All brain surgery is major brain surgery. But if we could do things like this without having to do surgery, that would make things even better. And this is a machine called focus ultrasound. And it's now been approved for making lesions in the brain, unilateral essential tremor. But I just want to show you that I'm doing studies, and I'll show you the data in just a little bit. So they go into an MRI scanner, and there's the brain. And what happens is they do focused ultrasound. But they take a spot on the MRI, and they put bubbles and ultrasound in to that very small spot. And what you could also do is, so I want to show you something. So I was in Madrid. Let me just shut this because this was unbelievable. This is going to be like another miracle. I'm in Madrid and they're doing this procedure on someone with essential tremor. And she's being wheeled down in the gurney. I'm sitting right there and she's going like this. And what they ask them to do, and this is very common, is there's a spiral and they ask the woman to draw inside the spiral. And so that's where she is at baseline. She goes into the scanner. They do this focus ultrasound for 10 minutes. Better, but not perfect. So they say, well, let's put her back in for another 10 minutes. Tremor completely goes away. And if it stays like this for 6 months, it stays like this forever. So what we're doing, actually, is this is with Dr. Jose Obesa, my dear friend from Madrid. This is another great thing about being scientists. You go to unbelievably great places to do your science with unbelievably great people. And so this is an animal in which this bright spot here, we opened up the barrier between the brain and the blood. And this compound that we normally cannot get into the brain can now get into the brain. So there are a lot of therapies that are powerful, but they don't really get into the brain very well. This is a new technology called focus ultrasound. We just got a grant to study this. And we're now able to, without making an incision into the brain, get into the brain with and deliver specifically potent therapeutics exactly where you want them. So we're incredibly excited about that. Now I want to talk briefly about Alzheimer's disease. Alzheimer's disease is the big white elephant in the room. We now believe, and I'm not going to have a chance to discuss this very much, that Alzheimer's disease may start in the stomach, in the gut, because there are some bad proteins that clump in the gut. And in fact, we believe, there are many people who believe it gets from the intestine and then spreads up to the brain and then spreads throughout the brain. And there are a lot of efforts now being made to prevent this spreading of this bad protein called alpha-synuclein. It's basically a protein that's normally in the brain, but for reasons we don't quite understand, it clumps. And it's sort of like your garbage disposal. If you just put a little bit down the garbage disposal, it'll clear it out. But if you put all this bad stuff in the garbage disposal, it'll eventually clog. That's sort of what we think happens both in Parkinson's disease and also in Alzheimer's disease. So here's a picture of Dr. Alzheimer. And this is a picture of Auguste D., the first patient who ever was described with Alzheimer's disease. This was in 1903. And we know that when Dr. Alzheimer looked at the brain of Auguste D., he found these abnormal features, what we now call plaques and tangles. And here is a neurofibrillary tangle from her brain. Now, the problem we study sometimes is we don't have models that are reliable for testing experimental therapeutics. And the other thing about Armageddon is that by the year 2050, there are going to be 125 million people with Alzheimer's disease. So we need to do our job, and we need to do it quickly and to do it with great rigor and intent. So, and especially low and middle income families, countries that are totally underserved. So we got to do something. This is the pathologies that I talked about with Alzheimer's disease. We have a compound called beta amyloid, which is outside the cell. We have tau, which is inside the cell. And then we have brain atrophy that occurs over time. So we got to get therapeutics that will address this. And so we have recently created a monkey model of this tauopathy. I'm just going to briefly go over it because it has changed the landscape of Alzheimer's research preclinically. We injected a virus, not a virus like we get, but a virus that's made in a laboratory, into a place where Alzheimer's disease starts, and it's called entorhinal cortex. And I'm using my pointer to point it out here. So this at 3 months, we see all this red and a couple of greens. All this red is a cell that is about to become a neurofibrillary tangle. And this is at 6 months, you can see much more green. And because of this model, we have biotech companies knocking on our doors. Takeda, we have a contract. Genentech, we have a contract. This is the entrepreneurial spirit that I want to emphasize for biodesign and for the NDRC. And because they have the resources to really push the needle, and we have the experience and the expertise to take those resources and apply them in a way that will help patients with Alzheimer's disease. And by that, helping them will reduce the caregiver burden, and it'll be a lesson for young students going forward. All our missions can be addressed in these particular studies. So how can you help? We're all in this together. So please, if you're a patient, with discussion with your doctors, please consider participating in clinical trials. I can't tell you how many clinical trials don't have a single patient enrolled. It's just amazing. And then go on the internet, look at where there are patient registries, if you're a patient or not, because it's very common to get patients to enroll in these registries, but we need the spouses and the control material to compare things to. So if you're a patient or not, donate specimens for various registries. So with that, I'm going to close, say thank you. It's been an honor and a privilege to be speaking with you today. And let me tell you something, I love doing it. So if you enjoy yourself, tell Kerry that to invite me back next year, and I'd be happy to do it again. Thank you very much.
Wow.
That was great. I guess we will move on to some questions now.
You can either put them in the chat or state them out loud.
So, hey, Jeff, do you want to just talk a little bit about how you get these, you make these stem cells by adding the Yamanaka factors? How do you encourage them to become neurons? How do you get them to
So
do
what
it?
happens is, the first thing you got to do is drive them. So you take the blood and you have these 4 factors, and that drives the cells, the blood cells, back to an embryonic stem cell without needing the embryo. And then you put factors that normally cause a cell when it's born to become a neuron. You put them back, and so you drive them forward. And then you do a little special sauce, and you give them a special cocktail to make them not just cells that make dopamine, but cells that make dopamine just in the right place. And so that's how we do it. And I got to tell you, something that would be 20 years ago thought to be impossible is now routine.
Wow.
Yes, I would like to make a comment and just amplify what Dr. Kordower has already said, that Alzheimer, when he first described this one patient and this one brain from this one patient more than 100 years ago, the plaques and tangles that he described then are still being used very heavily in Alzheimer's research. And there are some studies, there are many studies really, that are working not on these plaques and tangles themselves, but on precursors of the plaques and tangles. And Jeff, do you want to say something about that?
Well, we're trying to, right. So there are these precursors to the plaques and tangles, such as the beta amyloid precursor protein, and also these pretangles that stain for something we call AT8. And that allows, if we can get earlier and earlier and earlier in the disease process, we have a much better chance of preventing them to ever evolve into full-blown tangles and full-blown amyloid plaques. And that's a big effort that is undergoing by many people in biotech and academia. I can tell you there's this big controversy about Biogen and aducanumab, which was just recently approved, that is based upon removing amyloid from the brain. It can remove amyloid from the brain, but doesn't seem to make people better. It's a little bit of a controversy. I'm a tau guy. I think these tangles are much more important in terms of someone becoming demented, and the ability to prevent the tangle formation is a huge win if we can do this.
Jeff, there's a question from Peter Williams in the chat. It says, does breaching the blood?
I'm online, so I can.
Oh, yeah, okay. Please
Okay,
ask.
thanks. So you talked about breaching the blood-brain barrier, which I assume was put there by nature for a purpose. And so my question is, what negative consequences could there be from breaching that barrier?
That's an excellent question. There are no negative problems here, because what we've also demonstrated, and it's not just been demonstrated by us, is that by this is a transient thing. You open it up, and by 24 hours, it's back closed again. And you can do this repetitively, time and time again, and there doesn't seem to be any deleterious effects. This has been done in patients with Alzheimer's disease already, with some reported benefit. So we know in humans, it has passed muster in terms of the FDA, in terms of safety and tolerability. And so I don't think that this is going to be a problem going forward.
Okay, that's great. Thank you.
Dr. Kordover, Shelley asked in the chat, are you looking for patients for these tests right now?
Yeah, and I'm online as well. Just in short, within an 8-month period, my mom went from not being able to sit down because she just wanted to stay busy. She loved to be active. And with 8 months, now she can barely get up off the couch and she's on a walker. And the videos that you showed in the beginning with the lady doing the finger touches, my mother is so slow to react. And also trying to watch her walk on a walker. I got very emotional when I saw what the stimulation of the brain can do. And if you're looking for any person that wants to do testing, I know she would jump at the chance to get this done.
Well, first of all, let me tell you, I'm sorry that you're going through this with your mother. It's heartbreaking. And but let me just point out, first of all, one thing. I'm not a physician, so I cannot give medical advice,
Right.
particularly to someone individual. But the one bit of medical advice I will give is go see a movement disorders expert or go see a neurologist who's expert in these things so they can guide you. I gather you're in the Arizona Avenue. I could recommend Chuck Adler at Mayo, or Holly Schill at Barrow, both outstanding movement disorder experts, and they'll be able to tell you what is available and what's appropriate for your mom. And I
Yeah,
wish you the best.
She does have an appointment next week with the Barrows movement doc.
Okay.
So,
She'll get great care there.
okay, great. Thank you, Doc. Thank you.
Dr. Kordower, D asks, is there a special diet you recommend to counteract the possibility of brain disease starting in the gut?
Very interesting you say that, because I just submitted what's called a UDOL application, it's a large center application built around the gut-brain axis. In part, so there are 100 trillion different bacteria in your gut. And so this is a very complicated area. But there are these, and what I'm about to tell you now is totally unproven. It's actually what we're trying to study. That there are these prebiotic bars, chocolate bars, and they they are meant to normalize the bacteria in the gut. We know in patients with Alzheimer's disease, in patients with Parkinson's disease, they have what's called a leaky gut. The bacteria are all screwed up, and things leak out of your gut into the abdomen that shouldn't happen. And we believe that's reversible with these prebiotic bars. This is experimental. It's not ready for to be taken. So I really can't give you a good scientific answer to your question, but I hope to be able to give you a good scientific answer to your question as we go down the road.
How about related to that? What do people recommend as a just good general diet for the brain?
Well, I mean, you want to have, I think if you have a heart healthy diet, you're going to have a good brain diet.
Right.
So you want to keep things low in carbs. And I can just tell you, I lost 32 pounds, and I was/am a diabetic, but my diabetes has basically gone away. And when your diabetes goes away, your brain function gets better. And so being healthy with regards to your diet, I think, is very important. The other thing that I would recommend, and again, this is sort of against my I'm not a doctor kind of thing, but speak to your doctor. Exercise is incredibly important for health, not just physical exercise, but mental exercise. And so doing crossword puzzles, reading books, if you can't run, walk under the guidance of your doctor. And I think that is one of the most incredibly important therapeutics that has been proven to be helpful in patients with degenerative diseases.
I'd like to ask a question. We joined late. This is Penny Post. And just a year ago next week, my husband was diagnosed with primary CNS lymphoma. He was treated at Mayo Clinic, and the tumor is gone and has stayed gone. But one of the things, we came late, so I missed some of the first part of your lecture, but one of the frustrating things for us making some decisions is that he's 81, and there are almost no studies that include anyone older. There's one study with one person at 75, and we were told the reason they won't let us participate in a study at Mayo because he's too old and he could die from something else, so they don't want the odds to go down. So it's very frustrating trying to figure out what to do and how to do it. And
Well, I understand your frustration. That's not a disease that I'm really expert in, but I am expert in clinical trials. And I can tell you that, like, the Parkinson's trials are probably going to be limited to patients who are 45 to 70, because people who are older than that, again, if you pass away, then you've invested time and money, and it really messes up the data that you're collecting. Also, if you have trouble making it for your appointments, that messes up the data. So this is very common in clinical trials in general. I'm sorry about your frustration, but again, I would relate that frustration to your physician and ask what solutions he or she may be able to provide.
I don't have a lot of suggestions, but
Unfortunately, that's true for a lot of different things.
fortunately, he's doing really well. The tumor is gone and has
Well,
remained.
that's a great outcome.
That's
So-
a great outcome, and except for a few memory issues that have remained, which are definitely related to the tumor, we've done well, and certainly we did a lot of mental therapy as well, and that has, as well as exercise, been fabulous. So I definitely recommend it.
I agree.
What other questions do we have?
Well, I see someone put up there, what's the difference between a PET scan and a DAT scan? So a DAT scan is done by a machine called SPECT, single photon emission tomography. PET scan is a positron emission tomography. It's two ways to look at very similar things. A SPECT scanner uses iodine to label the chemical. So the pictures I showed you, those images of red and green, those are all PET scans. And they use a compound, like a dopamine, where they attach, this isn't going to be that important, a fluorine residue to it. And that's what makes it light up on that machine. A SPECT scanner uses iodine to basically do the same thing. So the DAT is a dopamine transporter. It's inside dopamine cells. It will show you the exact same picture under this circumstance for Parkinson's disease. So they're very similar. The DAT scan is FDA approved. The PET scans are more, they're not that they're experimental, but they are, they're not used as often.
We have time for some more questions.
Dr. Kordower, I would like to, you talked briefly about things that happen in the brain before tangles actually form and before plaques actually form. And I would wonder if you could make some speculation, and it would just be speculation, I understand, about how long these processes that precede plaques and tangles have been going on in the brain before someone reaches a stage where there's a clinical diagnosis.
Sure. Well, we believe that what we call the prodromal state, and I'll make this a more global answer because it applies to Parkinson's as well as Alzheimer's disease, that you probably have pathology starting in the brain decades before clinical symptoms appear. For like Parkinson's disease, you have to lose 80% of your dopamine before you start to see symptoms. We don't know really about as definitively with Alzheimer's disease. I can tell you, I published a study a couple of years ago in which, so for Parkinson's disease to be diagnosed, you need to have two of the following four cardinal symptoms. You need to have slowness of movement. And then one of the following three: postural instability, you fall a lot, resting tremor, and also rigidity, and rigidity of what we call a cogwheel form. And so I had a subset of brains from patients with Parkinson's disease, so they're diagnosed. Then I got a set of brains from patients who had one cardinal sign of Parkinson's, so they could not be diagnosed with Parkinson's. And this bad protein I talked to you about it a little bit, it's called alpha-synuclein, that clogs up the garbage disposal. It was rampant in those patients. So we know the pathology starts very, very early. And in a sense, if there's a long window between when the pathology starts and the symptoms occur, if there's a way we can get a therapy, two things. We need to be able to say this person is going to get Parkinson's disease, and we have a therapy that'll slow this pathology. You have a long window for which the therapy can be introduced and hopefully will have a significant impact upon the patients who would eventually get Parkinson's disease. So there's an upside and a downside to it, but we still got to do a lot of work on the biomarker side to be able to say, take a blood test and say, oh, this person's going to get Parkinson's disease. Let's start a treatment, 10 years before he's going to get it. And also develop these therapies that we know can slow this progression of the disease.
Great lead into the next question, which is on, is there a project at ASU that's trying to find biomarkers for these sorts of disorders? And so that all the person would have to do is speak into their phone and tell if their larynx is affected by disease or not, for example.
So yes, we are doing a lot of work in the biomarker field. I can tell you this model that I showed you towards my end of my talk, we have a big battery of proteins that we look at in blood and CSF, and they are proving to be excellent biomarkers. We now have to validate them in patients with Alzheimer's disease to see whether the model mimics what we see, and we're doing that. And I'm not sure exactly what you mean, but I could tell if your larynx is affected, although, hypophonia, Parkinson's patients talk very low and they have very little speech. And so those are the symptoms of Parkinson's disease. That could be an early sign and also an early biomarker as well.
Dr. Kordower, I had one question for you. That is, with incredible talk, but with all the work that you're doing, what could help accelerate your research at the Biodesign Institute?
Well, what's going to accelerate my research is, first of all, I'm going to hire some wonderful, not hire, but have some wonderful undergraduate and postdoctoral fellows work in my lab. I'm going to inspire them. We're going to work hard. I'm going to, as I'm trying to do, build a consortium of people in the neurodegenerative disease space in the Arizona community. We need help from our patients to go into clinical trials. Certainly, the knee-jerk answer is, if we had more money, things would be easier. That's always true. But, we're going to do great here, and we're going to make a big impact. So I'm still getting my feet on the ground a little bit, but we have some very talented assistant professors in our center that are making great strides, and I can't wait to get to work.
Do you want to touch on some of the differences between ALS, Parkinson's, and MS?
Sure. Well, ALS, amyotrophic lateral sclerosis, or Lou Gehrig's disease, is a disease where certain neurons in a certain area of the central nervous system, mainly the spinal cord, degenerate. And so they lose the axons, those processes that go out to the muscle. And also what's horrific is the motor neurons, those motor cells that control the diaphragm get affected, and eventually breathing becomes very problematic and you have to go on a respirator. It is 10% genetic. Most of it is non-genetic, and it's a very, very tough disease. Excuse me. I was thinking of actually having a ALS person in the center, but my good colleague Bob Bowser is a world-renowned ALS researcher at BNI, and we're probably going to build that bridge at the Center for ALS. Parkinson's is what I told you. It is mainly a movement disorder, but also it's a cognitive disorder. People get depressed. And as we talk about the gut-brain axis, one of the day-to-day biggest complaints from patients with Parkinson's disease is constipation, unrelenting constipation. And it's probably due to this change in the gut microbiome. And while ALS and Parkinson's are relentless, MS, multiple sclerosis, is a white matter disease. This is the area of the axons, not that cell body. I took those white parts of the cell that I showed you. And there's a large part where they get worse and they get better and they get worse and they get better. And so we call that remitting and relenting multiple sclerosis. They are fundamentally different diseases, although ALS and Parkinson's all have this misfolded protein that screws up the garbage disposal.
Okay, I think that's all the questions.
Thank you. It was great.
Carrie, you're next.
Well, if there's no further questions, let me thank you for a great talk, Jeff. We do have an upcoming event, the Arnson Grand Challenges lecture in early November. We're having Zach Rogers come speak to us about supply chain issues. I'm sure you've all heard in the news the issues that we're facing with lack of supply of microchips that's affecting car production, appliances, home improvement materials. So supply chain is really a critical issue brought to the forefront thanks to the pandemic. So Zach will be speaking on that topic soon. We strongly encourage you to sign up for that event. We hope you take our survey. We're going to send out a little email with some survey topics that you might have input on. We'd appreciate that. And at this point, I'd like to introduce Elizabeth Munkle, our Director of Development for the ASU Foundation. We hope to see you at the Arnson Grand Challenge and subsequent SIP of Sciences.
Thank you, Steve. As Steve mentioned, my name is Elizabeth Munkle, and I have the great pleasure of working with the Biodesign Institute on behalf of the ASU Foundation. Tonight, you got a glimpse of the amazing work taking place every day at Biodesign. We would love for you to join us in making this work possible. One way to accomplish this is to join our Pioneers Circle. The Pioneers Circle is comprised of science enthusiasts, curious knowledge seekers, and committed philanthropic supporters who invest in Biodesign's groundbreaking research. Your gift of $1,000 or more annually empowers our team to take on the most pressing challenges to our health and environment. Philanthropy plays a unique and vital role in our work by allowing our researchers to explore new and untested frontiers of disease detection, drug therapy, and sustainable materials. To learn more or to join, please contact me or visit the website in the chat box. Thank you.
Thank you, Elizabeth. Thank you again, Jeff. Thank you to tonight's events team. We look forward to seeing you at another event. Have a very good evening.
Thank you.
Bye. Thank you. Thanks for joining us.
Bye,
Have a great
everybody.
night.
Thank you and good night.
Thank you. Bye now.